Happy Friday. I imagine that so many of you on this day are really breathing a sigh of relief as you head into spring break. And I hope that all of you have something really fun planned. And if you don't, then you should plan it right now and make sure that you do get a break and maybe take your studying on the road. I often find that it works much better to study in new locations. You learn it more because you associate it with some new environment. So take your micro with you and go on the road and find something fun to do. I have only just a few announcements, and these announcements are really looking forward in time, so not anything that you need to worry about probably until after spring break. But your seventh homework assignment will be due on Tuesday, March the 20th, and that will be followed up the next week by your third exam. And this exam will be perhaps one of the most difficult exams you've encountered, but on the flip side, you are allowed to use your poster. So if you do a good job of really making a good poster that has on it both metabolism and gene expression, if you would like, you can bring that with you. And speaking of bringing things with me, my cat Gabby has decided to come to the vodcast with me tonight. But you can bring that poster with you, whether it be on a bed sheet or a curtain or whatever it is that you have decided to use to make your poster. Now, there are a few rules on the poster. One of those rules is that you may have no blocks of text, so it needs to be a picture. I want it to be in picture form. It also needs to be of your own creation. That is, I don't want you to make copies from something that was in a book or something that I did and photocopy those things and put it onto your poster. I want you to create it and build it. And it does need to be a single sheet, right? It can be on a sheet, or it can be on a poster board, or it can be on anything you'd like, so long as it is a single sheet and goes by the other rules there. Okay, when we left off last time, we were totally stoked on gene regulation, and we had been talking about the way in which DNA binding proteins can also be allosteric proteins. We had namely ended with repressors, and we had talked about how effector molecules can be either co-repressors or inducers of repressors. So likewise, you may have even thought this before the end of the hour last time, that also means that activators can also be affected by effector molecules. And here's an example of that where we can see an activator that normally doesn't bind to the activator binding site, but if it is bound to its effector molecule, which is clearly an inducer, then it's going to allow it to bind to the activator binding site. Now, wait a minute, that sounds sort of confusing, doesn't it? Because here, an inducer is allowing the DNA binding protein to bind, whereas with repressors, when an inducer was bound, it kept it from binding. But remember, it's all about the downstream genes. So depending upon whether or not those downstream genes are upregulated by the binding of an effector or downregulated, that's what dictates whether we call this an inducer. So remember, because an activator does exert positive regulation, we recognize that it would be called an inducer when it allows the activator to bind. So it's sort of an opposite sort of relationship there. Okay, so we now have summarized two ways in which genes can be regulated. The first way is via the promoter sequences and the differences in promoter sequences. The second way is modulation by DNA binding proteins. And lastly is a sort of complicated mechanism, and that is something that we call specialized sigma factors. So you better get that picture all labeled up. But specialized sigma factors is a third and final way that we'll talk about to regulate gene expression. Now, I don't know if anybody's remembering, but I'm hoping that some of you are certainly remembering the very promiscuous sigma subunit of RNA polymerase. This is the subunit, remember, that allows the RNA polymerase enzyme to bind to and recognize the promoter sequences. So, sigma factors can come in all shapes and sizes. We know that some sigma factors, or many sigma factors, are the type that recognize TT-GACA and TATA-AT in the promoter. However, not all sigma factors recognize that specific promoter sequence. Some of them recognize new and unique and novel protein sequences, or promoter sequences. Oh my goodness, I'm going to have to get rid of my kitty. She is way too excited about this. Okay, she has her feelings a little hurt, but I have now kicked her out of the room. So we do have some specialized sigma factors that recognize unique promoter sequences. They're not the typical TT-GACA and TATA-AT. So let me write out one unique promoter sequence. And it'll make sense why this might be a sigma, why this might need a specialized sigma factor, because it's not a typical gene that needs to be expressed all of the time. So here is the promoter sequence for that, and it's C T T G A A. That's the -35 sequence in this promoter. And then further downstream is the -10 sequence, which is, in this case, CCATA. Now you might be thinking, what the heck is N? That is a generic nucleotide, so it can vary what's in that position. So this is a promoter sequence that looks nothing like TT-GACA and TATA-AT, but in fact it is the promoter sequence that is upstream of genes that are required for heat shock. That is, when a cell is under duress, is the only time that these genes would be expressed. So that's when a special sigma factor will come in and recognize and express those genes. So the average everyday sigma factor won't be used for that. So that's the way in which heat shock genes are regulated. This is also the same mechanism that's used to regulate endospore forming genes, right? Only under very unusual conditions are those genes going to be expressed. So the production of specialized sigma factors is one way to regulate the expression of genes that are only needed under certain environmental conditions. That is for sporulation or for heat shock. Those are good examples of that. So I have a feeling this leaves you going, wow, I need a review of what are all those mechanisms that are used to control gene expression. So let's power through a quick review of those different mechanisms. We'll begin, of course, with variants in promoter sequence. The closer to ideal, the more frequently expressed. And of course, the second is modulation by DNA binding proteins. Repressors and activators. You can tell it takes me a lot of concentration to write with this pen. And then lastly is the production of specialized sigma factors that recognize only promoters that are upstream of genes needed in very unusual environmental conditions. So specialized sigma factors. For example, those involved with expression of endospore forming genes. So these are the three ways that we talked about for controlling gene expression, remembering that it makes sense to regulate gene expression because it's silly to make what you don't need, and a cell is very conservative in that way in only making what it needs when it needs it. Not with all things, right? Because certainly some things are always turned on. Okay. Let's hit a practice question here, and as we go into this practice question, one of the things that we are going to see is that gene regulation can become even more complex. And you'll notice this right off the bat, because before we talked about the Lac Operon, where we said it would have a promoter, an operator, and genes 1, 2, and 3. But now what I'm showing is that not only do certain operons, such as the Lac Operon, have an operator, but they also have an activator binding site. So spec that, right? This is crazy. It means that this particular operon is controlled not only by a repressor, but also by an activator. Not only by potentially an inducer of the repressor, but also maybe an inducer of the activator. So this is intense regulation. So let's see if we can practice this question. If an activator is bound, okay, so an activator is bound. So that means that we've got this activator and it binds. That, of course, exerts positive control. That means it's making more likely the expression of genes 1 through 3. Now, an inducer is bound to a repressor. So remember that that means that the repressor normally binds to the operator, but because there is an inducer present, it's going to alleviate that repression. So plus the inducer is going to lead to the formation of a repressor that no longer represses. That is, it has been induced. So it's not going to bind the operator. So when we look at that, we recognize that there's no repression of expression, but there is activation of expression. So genes 1, 2, and 3 are transcribed. RNA polymerase happily binds and zooms on down that transcript. So we can answer this question as C. Genes 1, 2, and 3 are expressed. But let's see why A and B are wrong. The repressor binds to the operator. No, it doesn't, right? Because it's been induced, so it's not binding. And then B, the repressor binds to the activator. Well, that's just bunk, right? That doesn't happen. The repressor binds to the operator, not to the activator binding site. Okay, so with that practice question in mind, we can now zoom along to talk about complex gene regulation. And that's what we're seeing here. That is when a construct is under control of both an activator binding site and an operator. This is something that we see on the Lac Operon, and we see that one of the forms of regulation of that operon is something called catabolite repression. Now, when I think of catabolite repression, I think of the ski team, and that's because I think of the way that they eat. You know, if you take the ski team to a buffet, the first thing that's going to happen is that they're going to go and they're going to exhaust the buffet of all of the things that they like. And then they're going to pause and they're going to be like, I'm still hungry. And they're going to go back to the buffet and exhausted of all the things that they don't like, right? So this is very much what we see with E. coli as well. It's going to begin by digesting the sugars that it likes the most, aka glucose. Then it's going to pause and it's going to move on to lactose, the sugar that it doesn't like quite as much, the disaccharide that it has to break down. So it'll pause and move on to lactose. And that's what this graph is showing here, is a preferential use of glucose, but then the use of lactose secondarily. This is called dioxic or two-phase growth. First on glucose, then on lactose. So looking at the graph, we see bacterial density on the y-axis and time on the x-axis, and the first curve of growth shows growth on glucose. The second curve shows the pause first, and then it shows growth on lactose. So consuming its preferential sugar first, pausing, and then consuming the lactose disaccharide that it doesn't like as much. So this is the okay, I've got to try the joke, even though I know you guys are too young and you're not going to get this. But this is the E. coli growth curve. So this is E. coli. This is E. coli on glucose. Are there any questions? Yeah, you probably don't even remember that. This is your brain on drugs, but whatever. So let's go ahead and talk about what this means. And I think the best way to do that is to refer to something that you're doing in lab. Remember the KIA tubes, or the Kliegler's iron agar, where it's packed full of both glucose and lactose. Now, what that means is that the E. coli that are put into that KIA tube are going to preferentially use the glucose first. They're going to ferment that, turn the tube yellow, but then they're going to move on to the lactose. They're going to then ferment the lactose, maintaining the yellow color in the tube. So what this means, let's think this through, right? If E. coli is preferentially able to utilize glucose and not use lactose until it's done with the glucose, doesn't that mean that the presence of glucose is somehow inhibiting expression of the digestion genes that digest lactose, right? Think about that. That's what catabolite repression is. The presence of one metabolite is repressing the expression of the genes needed to digest the other metabolite. So this is pretty crazy. So what we're recognizing is the ability of one metabolite, glucose, to repress the expression of the enzymes that digest lactose. This is given that term catabolite repression. But what's interesting about this is you might be visualizing the Lac Operon and you might be thinking, oh, well, that's easy. That just must mean that glucose acts as a repressor and binds to the operator. Not so simple as that. It turns out that glucose is kind of a wimp. It doesn't do its own repression. It's kind of got a hired hitman to do the repression for it. It is not the actual molecule that exerts control on the Lac Operon. Instead, there's another molecule called cyclic AMP that is responsive to the presence of glucose, and it is what regulates the Lac Operon. This molecule is called, well, it's called cyclic AMP, and it's a secondary messenger in cells. Maybe you've heard of it before. Cyclic AMP, though, is reciprocally regulated as compared to glucose. That means it's inversely related. So when glucose is high, cyclic AMP is low. When glucose is low, cyclic AMP is high. So we recognize that in a cell that has exhausted the glucose supply, suddenly this secondary messenger cyclic AMP is going to go way up, and that's going to exert control on the Lac Operon. So glucose is not sensed directly. Instead, cyclic AMP, a secondary messenger, is going to be sensed. So we'll look at how it's sensed. First off, if cyclic AMP is low, that must mean that glucose is high, right? If glucose is high, that means that the expression of the lactose degrading enzymes is inhibited. So when cyclic AMP is low, the degradation of sugars other than glucose is repressed. On the flip side, if cyclic AMP is high, then that means that glucose is low. That means, okay, I'll go ahead and digest those sugars that I don't like as much, aka lactose. So then the enzymes needed to digest the lactose are synthesized. So this is getting hard to picture, isn't it? So this means that I think we need to go on and actually draw the Lac Operon so that we can visualize how it is that cyclic AMP regulates expression of the operon. We've got to put this together with what we already know in terms of how lactose regulates expression of the Lac Operon. Remember how it was an inducer to a repressor. So this is how gene regulation starts to get very complex. So we'll recognize that the Lac Operon, looking at this picture, is regulated by both an activator binding site and by an operator. So it's under control of both. That means that in order for all systems to be go, the repressor can't be bound to the operator. That is, it needs to be denied in order to allow full expression. But also, we recognize that in order for full expression to happen, an activator must be bound to the activator binding site. I like to think of this as a two-switch system. You guys have some of those lights at home, I bet, where you have to turn it on at the wall first, and then you can turn it on at the switch. I have one. I have one here. So this one has to be turned on at the wall, and then it can be turned on at the switch as well. So I've got this little switch here, right, that I can turn on the light. And as long as it's turned on at the wall first, it can be turned on at the switch and it'll go on. Well, this is what's going on the Lac Operon as well. If the activator is bound to the activator binding site, aka the wall switch, then that switch is on. But also, we have to know that the repressor is not bound to the operator in order for all systems to be go. So this is a two-switch system. So we can now recognize each component of regulation, beginning with the activator. So the activator is something called catabolite activator protein, or for short, we call it CAP. The activator binding site, of course, is the CAP binding site. But we have to think about what will make cap bind in order to turn that switch on? And it turns out that's where cyclic AMP plays its role. So cyclic AMP is going to induce the binding of the catabolite activator protein to the activator binding site. So we recognize then that at the operator, well, that's going to bind, the repressor is always going to bind unless lactose is present. And remember how lactose serves to induce expression of the genes by keeping that repressor from binding. So in order to fully understand this, let's think about our KIA tube. And remember, when you stab down into the KIA and spread on the top there, the first situation that you have in the KIA is that both glucose and lactose are high. Okay, so if both glucose and lactose are high, then we know that E. coli is going to want to eat glucose, right? It's first going to decimate that glucose before turning to the lactose to start to degrade that. So in this situation, if glucose and lactose are both present, well, first off, we know that lactose is there to induce the expression at the operator. So a derivative of lactose will serve as an inducer for the repressor, binding to that repressor and causing it to be denied from the operator. Well, that means that one switch on our two-switch system is on. That is, the switch at the operator is on. However, notice that glucose is present. So that means that cyclic AMP is low, right? If glucose is high, CAMP is low. If CAMP is low, it's not binding to CAP and it's not inducing expression at the activator binding site. So the wall switch is not on, right? And we don't have all systems go. Only one switch is on here. But what about when all the glucose gets eaten up and there's only lactose present but no glucose? Well, then that means glucose is low, meaning cyclic AMP is high, meaning that cyclic AMP will bind to CAP and activate or induce that activator to bind to the activator binding site. Lactose is still present, so it's still serving to induce the repressor. The second switch is on, and we now get expression of genes 1, 2, and 3. The Lac Operon is fully transcribed. This can be a confusing system, and a lot of the reason that it gets confused is because people tend to think that there's some relationship between what's going on at the operator and what's going on the activator binding site. But in fact, that's not the case at all. In fact, the activator binding site just responds to the presence of glucose. If glucose is there, then CAP doesn't bind. If glucose is gone or mostly depleted, then CAP does bind. So this is just responding to the presence of glucose. Now, the operator is just responding to the presence of lactose. If lactose is present, then this is induced and there's no binding of the repressor. If lactose is absent, then the repressor binds and blocks. So if lactose is present but glucose is absent, then that's when we get the full transcription of the operon. Let's do a practice question so that we feel comfortable drawing the Lac Operon. And my suggestion is, for study purposes, I want you to sit down and draw the Lac Operon under every circumstance you can think of. Draw the Lac Operon when glucose is high and lactose is low, when lactose is high and glucose is low, when glucose and lactose are both high, when glucose and lactose are both absent. Draw the full operon in each one of those conditions. Let's practice one of them. We want to know, in this case, what is the appearance of the Lac Operon when E. coli is growing on glucose salt agar. Here's the recipe. Remember, it's got a bunch of salt and glucose. Not surprising, right? But notice that that means that glucose is high. So let's jot this down. Glucose is present or high. Lactose, completely absent. Nobody sees lactose in that recipe, right? So lactose is completely absent. So now let's draw the Lac Operon. Remembering that in its full glory, it has both an activator binding site and an operator. So we'll draw the activator binding site upstream of the promoter. Then, of course, the promoter, followed by the operator. I'm just going to label it O for short. Now we know that downstream then are genes 1, 2, and 3. So in this case, we recognize that glucose is high. Okay? If glucose is high, what does that tell us about cyclic AMP? Cyclic AMP will be low, right? So it's low. So that means that CAP, catabolite activator protein, has no inducer. Cyclic AMP is not around to bind to CAP to induce it, so it is denied. That means that the wall switch is off, right? It's not turned on at that switch. However, we know that the operator uniquely will respond to the presence of lactose, and in this case, lactose is absent. So if we look at what's going on there, the repressor has no inducer because lactose is absent, and so the repressor binds. Wow, this is like no switch is on, right? This is when glucose is present, lactose is absent. There is absolutely no way that those genes 1, 2, and 3 are going to get expressed. They're all systems no-go. So you can start from here to draw the Lac Operon under every other condition that you can think of. Here we've done high glucose, lactose absent, but you could do the opposite, right? High lactose, glucose absent. In fact, we kind of already did that one. But do it with both glucose and lactose, both or only lactose and only glucose, right? You can think of all the combinations that you might draw the Lac Operon. So knock yourself out with that, but more than anything, have a wonderful spring break, and I can't wait to see everybody after the break.